Hello, this is a MOOC presentation as a part of Basics of Extracellular Vesicles. I'm Suchal Jang from Crafting Research Center, University of Gothenburg, Sweden. The topic of this presentation is isolation method of extracellular vesicles focused on density gradient. It will start with a basic background of density gradient and will continue how this method is applied to EV isolation. Density gradient is a method that isolating or purifying the particles based on their size or density depends on types of density gradient. This method is usually coupled with ultracentrifugation isolation, which you may heard from previous book presentation. By using density gradient, you can get a pure vesicle population. There are two types of density gradient, which are top loading and bottom loading, according to sample loading position. First, I will talk about the top loading density gradient. To do this, make the density gradient like this, highest density on the bottom and the lowest on the top. Then load the sample on the top of gradient. Apply the centrifugal first by ultracentrifugation. After ultracentrifugation, you can see the separation of particles. Small size of particles such as proteins are still remain on the top. You may recognize from this picture that particles are separated by size. The speed at which particles sediments depends primarily on their size and mass rather than density. In this way, the faster sedimenting particles are not contaminated by the slow sedimenting particles. Important thing is that the particle that has same density but indifferent in size will eventually end up at same position if centrifuged long enough. However, long centrifugation can induce sedimentation of small proteins as well, so choosing optimal time is very important for successful separation. Next one is bottom loading density gradient. This method is also known as buoyant density gradient or equilibrium separation. It's the same as top loading. Make the density gradient like this, highest density on the bottom and the lowest on the top. But highest density contains the samples. Apply the centrifugal first by ultracentrifugation, and then you can see the separation of particles. Upon ultracentrifugation, particles of specific density float until they reach the point where the density is the same as the gradient media. In this density gradient, particles are separated solely by their density. Particle size only affects the rate at which particles move until their density is the same as the surrounding gradient medium. This rate of movement is also called buoyancy velocity. Therefore, sufficient time is required to get the correct density of particles in this method. This table shows various types of gradient medium. Each gradient medium has their primary use because they have their own characteristics. Among these, we will focus two different gradient medium, sucrose and iodixanol, because they are used frequently on the EV isolation. Sucrose is a disaccharide that combination of glucose and fructose as shown in the chemical structure. It has 1.59 gram per ml density, so can generate higher density than water, which has 1 gram per ml density. This table shows density of a mostly used molar concentration of sucrose. If you want to find the density of other molarity of sucrose, please visit this website. The iodexonal gradient is also known as optiprep gradient. Optiprep is a product name of iodexonal. You can buy as 60% optiprep with a 1.32 gram ml density. There are different types of iodexonal solutions which have different density. In case of EV isolation, iodexonal sucrose solution is commonly used. This table shows density of each percent of iodexonal when we use an iodexonal sucrose solution. More information can be found in the commercial webpage of OptiPrep. This slide is about difference between sucrose and iodexonal. Viscosity, osmolality, and effect on EVs are main difference of them. In general, sucrose has high viscosity and hyperosmotic. In addition, sucrose may affect the functionality of EVs, although it was used successfully in the past for functional study of tumor EVs. On the other hand, iodexonal has low viscosity and isoosmotic. In addition, provider says that it is non-toxic to the cells, which may not toxic to EVs as well. So why density gradient is needed for EV isolation? Important thing is that density gradient can increase the purity of EVs. From Van Dien's works, they have shown that relative protein level for particles is the lowest in the optiprep density gradient as compared with other purification methods such as ultracentrifugation, exoquick, and total exosome isolation kit. In addition, even marker proteins, ELIX and TSG101 are enriched when optiprep density gradient was used, as compared with other isolation methods. These results suggest that optiprep gradient may be helpful to get rid of contaminant proteins and increase the purity of EV isolates. Another point of using of density gradient is that we can isolate the subpopulation of EVs according to size or density. In this study, authors showed that different density of EVs can be isolated by density gradient. Here they use the sucrose density gradient with the two different loading positions, top loading and bottom loading. As I mentioned before, separating factor is different on those two methods. By using top loading, EVs are separate based on the size, but centrifuge long time, it'll end up at same position, so they got one fraction. However, by using bottom loading, EVs are separate based on the density, so they got two different density fractions. From top loading, particles are localized near the fraction number 4, which has even marker proteins. From bottom loading, two subpopulations of EVs can be seen near the fraction number 4 and fraction number 9. Both fractions have even marker proteins with a slightly broad distribution. Electron microscopy of two subpopulations of EVs reveals that low-density EVs are slightly bigger than high-density EVs. In conclusion, this study shows that by using density gradient, we can isolate the different types of EVs. Here I will explain another type of method for EV isolation called cushion-based isolation. This is very similar with the top loading density gradient, but slightly different. On the bottom, load high density medium such as 2.5 molar concentration of sucrose or 50% optiprep, and then overlay the low density medium. Finally, overlay the sample on the top. Here you can load large volume of samples, which is different with the top loading density gradient. Apply the centrifugal force. All the particles will sediment to interface of a low and high density medium. However, protein contaminants are still remain on the top. Difference from top loading gradient is that different size of EVs is not separate on this method. Instead, all the EVs will end up at same position. So using this method, you can concentrate your samples and can get rid of most of the protein contaminants. In summary, these are considering factors when we use density gradient. First, you have to choose a gradient medium, sucrose or iodixanol. Then have to choose loading position, top loading or bottom loading, based on your purpose of study. In addition, centrifugation time is very important. In case of top loading, if you centrifuge for a long time, particles will end up at the same positions. On the other hand, in case of bottom loading, if you centrifuge for a short time, particles cannot reach their correct density. Therefore, choosing appropriate centrifugation time is very crucial for successful purification. For more information, please find these references. They have done density gradient very nicely. This is the end of my presentation. Thank you for your attention and good luck with your experiments.